Measuring mesophyll conductance with the CIRAS-3 portable photosynthesis system will be presented by Dr. James Bunce. For this webinar, we ask that you keep your microphone on mute for the duration of the presentation and put any questions you have in the chat. Please keep your questions related to this topic. Once the presentation is complete, Dr. Bunce will address your questions. For any questions we don't get to, we'll be happy to answer them directly via email. If you have any unrelated questions, feel free to email me directly and we'll make sure they get answered. Now I'd like to introduce Dr. Bunce. Jim built his first photosynthesis system as a grad student 48 years ago and has been performing leaf gas exchange measurements ever since. He recently retired from a 40-year career with the USDA ARS in Beltsville, Maryland, as an environmental plant physiologist. His most recent work involves predicting impacts of global change on plant physiology. He's worked with many brands and models of photosynthesis systems, he'll tell you, over the years, and prefers the CIRAS-3 overall. So much so, in fact, since his retirement, Jim has been working closely with PP Systems with our development and engineering team, testing and developing instrumentation, as well as providing technical and application support for all of our customers. So I want to thank you for doing this again, Jim, and you can take it from here.
4 methods of measuring mesophyll conductance with the CIRAS-3 portable photosynthesis system. So what is mesophyll conductance? This gets a little confusing because the definition has changed over the years. And if you read the literature, you may get different indications of what that really means. The old definition, back in the 1970s and probably through part of the 80s, was mesophyll conductance was just synonymous with the initial slope of the A versus CI curve. So that was the old definition. The new definition, now mesophyll conductance refers to the movement of CO2 from the intercellular airspace, which is indicated as CI, to rubisco, which is, or CC, C at the, in the chloroplast, and specifically in C3 photosynthesis. The term mesophyll conductance is still used, but with a completely different definition when people are talking about C4 photosynthesis. So you have to realize that mesophyll conductance means different things to people who are interested in C4 versus C3. So I'm going to talk strictly about C3 photosynthesis. And this is just a simple equation. Mesophyll conductance equals A divided by the difference between CI and CC, where A is the net rate of CO2 flow, which is a CO2 assimilation rate. CI is the CO2 concentration in the intercellular airspace. And CC is the CO2 concentration at rubisco. And that's our big unknown. So that's what we have to, the trick of measuring mesophyll conductance is to get a good estimate of what CC is, the CO2 concentration at rubisco. And we do that by using a photosynthesis model, the Farquhar-Van Kammer-Berry model. So the question is, why do we care? Why measure mesophyll conductance? Well, the real reason is because it's another source of genetic or environmentally caused variation in photosynthesis. There's intraspecific as well as interspecific variation, and it's often correlated with maximum photosynthetic rates. It does respond to the environment, especially to temperature, but also to water stress. And it could cause genetic variation in leaf water use efficiency. So those are the reasons for being interested in it. Basically, mesophyll conductance affects the curvature of A versus CI curves. This is just an old figure taken from a paper we'll talk about later with the fluorescence methods. This is a Loreto et al. Paper, and just shows A versus CI with varying with infinite mesophyll conductance and decreasing values of mesophyll conductance. And you can see with infinite mesophyll conductance, you get a steep initial slope and it curves over fairly quickly. And as the value of Mesophyll conductance decreases, that curve becomes more linear and doesn't curve until much higher CI values. So that's fundamentally what Mesophyll conductance does, is it affects the curvature of ACI curves. And that is the first way of measuring it. So the method one for measuring it is just from the curvature of the A versus CI curve. There's a open access Excel utility published by Sharkey et al. That gives values for Mesophyll conductance from ACI data. It requires about 5 points in the Rubisco limited region of the curve. And the calculation assumes that Mesophyll conductance is independent of CI. And that's a somewhat dubious assumption. And we'll talk about that more in a minute. You can, to test as a way around just using the Sharkey utility, you can use CO2 ramping, which is available with a CIRAS-3, and get a dense set of data points. And you can then separate the A versus CI curve into segments to test for the constancy of GM at different CI. So those, hopefully most of you are familiar with CO2 ramping. This is just an A versus CI curve. The orange dots here are for points collected with traditional steady state measurements, and all the blue dots are connected with the CO2 ramping system in the CIRAS-3. And you can see that they pretty much define the same curve, but you can see the density of data points is a whole lot more with the blue than with the orange in a smaller amount of time. And then you can use the Sharkey utility over shorter segments of the CI range, and you can actually test whether Mesophyll conductance is the same at all levels of CI. That's actually only been done a couple times. So the question is whether it is constant in the Rubisco limited CI range. There's relatively little data. I published in 2018 measurements of that using several different methods of estimating Mesophyll conductance, and to ask the question whether it's constant over the initial part of an ACI curve. And in soybean and sunflower, it is constant as far as we can tell. But in another species, which was common bean, Phaseolus vulgaris, it decreases, Mesophyll conductance decreased strongly with increasing CI. And there is a little bit of literature that says that happens in other species as well. So that's the initial slope method. Methods 2 and 3, I'll put them together here just because both of them use a combination of gas exchange and fluorescence measurements. And the basis for this is fairly old, 1992 reference in plant physiology, which is given here by Harley and et al. And they developed two different methods combining gas exchange and fluorescence to estimate Mesophyll conductance. And we'll go through them separately. But first, on the CIRAS-3 fluorescence settings menu, the fluorescence measurements you need for these two methods of estimating Mesophyll conductance are both fairly simple. So this is just the screenshot of the settings menu. Just to point out a few things, you don't need to record the raw fluorescence. And the initial fluorescence measurement, it really doesn't matter what you put in here because you're not going to use it. And you don't need an initial dark period. We can just start out with lights on, and it really doesn't, your initial measurement is irrelevant. Your repeated measurement needs to be a measurement of the efficiency of photosystem 2. And right now, the preferred way of doing that is with a multi-pulse system. And you also do not need any concluding fluorescence measurements, and you don't need a concluding dark adaptation period. So it's a fairly simple menu. And so the only thing that really is important here is the multi-pulse duration and intensity. And just following the basic instructions in the CIRAS-3 manual, use a few steps of decreasing light intensity in step 1, step 2, step 3, and you can do up to 5 steps. Three is enough, but you decrease the pulse intensity at each step, and then and it gives you your efficiency of photosystem too. So it's a really simple simple fluorescence scenario to do this. So method 2, it's called the constant J. J refers to electron transport, photosynthetic electron transport. It's called the constant J method. And it uses this equation, which comes from that publication directly. And you can see that the J, the electron transport, is related to assimilation rate. Dark respiration, we assume is constant. And related to CI and assimilation rate again, and then mesophyll conductance. So what the method involves is finding a region where you can change CO2 concentration and yet J does not change. So that's why it's called the constant J method. So obtaining a constant J as CO2 varies is best achieved at less than saturating light and sort of mid-range of CO2 values. And if you target in there and vary CO2, you often come up with essentially constant J as CO2 varies. And that's what you're looking for this method. So what you do is then you go back to the equation I just showed you. You assume a value for mesophyll conductance, and then for a range of CO2, calculate J from that equation, and also get the variance of that calculated value of J using that equation on the previous slide. And the best estimate of mesophyll conductance is the mesophyll conductance value where the variance in J from that equation is at its minimum. So it's it's a little bit limited because you need to experimentally find a region of environmental conditions where J is constant as CO2 varies. But once you've found that region, then you can fairly accurately get an estimate of what the best mesophyll conductance is by finding the value that gives you the least variation in calculated values of J. All right, so there's some limitations. It's limited to CI values where J does not change with CI, as I said before. It's limited to light values, PPFD values, where J is constant with a change in CI. So it has sort of a limited flexibility in terms of the region of the response curve where you can use the method. And another limitation is it has fairly poor resolution when GM values are very high, which happens a fair amount at fairly high temperatures. We'll see later the GM goes up quite a bit at high temperatures. So this method doesn't work really well it doesn't resolve GM values very well at high temperatures. The third method is called the variable J method, again using fluorescence. And this uses a slightly different equation where mesophyll conductance is a function of the assimilation rate, CI, the photocompensation point, gamma star, and electron transport rate and dark respiration again. In this case, the trick is we can measure assimilation rate. The gas exchange will give us CI. We either use a modeled value for gamma star, or we can actually go and measure it, and the same with RD. The trick in this one is using fluorescence to calculate J. Now, fluorescence will do that, but the thing we need to know is we need to know the photochemical efficiency of photosystem 2 under non-photorespiratory conditions, which means either very high CO2, like 2,000 parts per million, or low oxygen, like 2%. And then we have the instrument tell us what J is. The instrument tells us what the photochemical efficiency is. And this constant we calculate when we know that J is being measured under non-photorespiratory conditions. So then the equation on the previous slide, then once we have a value for J, we can, the equation tells us how to estimate GM from A and CI. And you can do it over a range of CI, you can do it over a range of light intensities. So in that sense, it's much more flexible than the constant J method, because it can be used under a broader range of environmental conditions. Method 4 is from the oxygen sensitivity of photosynthesis. And Rubisco properties determine the photosynthetic response to oxygen. If the observed response to oxygen does not fit the photosynthesis model, then it tells us that the CI used in the model is different from CC. So what we do is we compare how well the model is fitting both the 21% oxygen measurement and the low oxygen measurement, and then we put in different values of Mesophyll conductance to find out what fits best for CC. So with the CIRAS-3, we measure assimilation rate in air, then measure it again in low oxygen using, ideally using 2% oxygen to nitrogen. And the reason for using 2% is so that the rate of dark respiration is not affected by that change in oxygen, since respiration is saturated at about 1% oxygen. So we use 2% just to be sure. Make two measurements, just assimilation rate at whatever CO2 we want, measure it in air, and then measure it in low oxygen. And from those, use the rate in 21% oxygen to calculate, depending on where you are on the response curve, either VC max or J at that temperature using the photosynthesis model, and determine whether that same value of VC max or J fits the data you got at 2%. And if the predicted value at 2% is bigger than the observed, that indicates that GM is finite, and we can figure out a value of GM that makes those two predicted and observed become the same. So recalculate the rates using different values of GM. So that can be done just by trial and error. But there's also an Excel calculator, which is, again, open access publication, and I'll give you that reference later. So that just speeds things up. It eliminates the trial and error approach. So it just comes quickly to a solution of what the GM value is, which makes the model fit both the rate of assimilation at 21% and the rate at 2%. Can be used at this oxygen sensitivity method, can be used at any temperature or light or CO2. But there's a big but, and that is that the same limitation to photosynthesis, either Rubisco, VC max, or J, needs to occur at both oxygen levels that you're using. And it turns out that this Excel calculator, which does this calculation quickly for you, is actually only developed for the Rubisco limited photosynthesis. So, so with the oxygen sensitivity method, using that calculator, it works fine if you're in the in the CO2 limited, or I mean the Rubisco limited part of an ACI curve. And then it very quickly comes up with a solution of what mesophyll conductance value fits both the 21 and the 2% oxygen measurements. So this is a reference to the method, which is an old paper of mine, 2009. And then this second reference here is the calculator. It's just an open access Excel calculator. As I said, it only works in the VC max limited region of an ACI curve. So if you're operating it, if you're trying to get a mesophyll conductance estimate up at higher levels of CI, then you have to go back to sort of trial and error, swapping in and out mesophyll conductance and recalculating the assimilation rates with the model at 21% and 2% and swap in and out mesophyll conductance values until those two estimates agree with each other. Okay, so sort of in summary here, the variable J method and the oxygen sensitivity method are the most generally useful. And by that I mean they work well at a range of temperatures or light intensities or CO2 concentrations. The variable J method does require the fluorescence, but it's a fairly simple fluorescence measurement to set up and use. The oxygen sensitivity method requires a 2% oxygen and nitrogen mix to be piped into the inlet air supply. But that's cheap stuff, and you just buy a small bottle of it and it lasts a long time. There is another way to do mesophyll conductance measurements, and that's with online carbon isotope analysis, but that's very cumbersome and extremely expensive instrumentation. So, so we have 4 other alternative ways of doing this. So the carbon isotope analysis is a 5th method, but it's not practical for most people. We can open it up to questions.
All right. Well, first, thank you, Jim. That was a terrific presentation. And we always appreciate your efforts. So we'll open it up to questions now. So if anyone has any questions, again, just feel free to place them in the chat, and we'll go through them here with Jim. So Jim, we've got our first question here. Can the high density ACI curves be combined with fluorescence to determine mesophyll conductance through the variable J method?
I don't see any reason why not. Yeah, I have not done that. Yeah, let me think about that. I've not used fluorescence with the CO2 ramping, and the reason I haven't is because the pulses of real high light with the fluorescence measurement temporarily mess up your leaf temperature, so that and I've always been afraid that that was going to affect what I'm getting for assimilation rates or CI values, especially when doing rapid CO2 ramping. I think probably it would be okay if you slowed down the ramping so that there was a longer interval between the longer time interval between the successive measurements of assimilation rate, but I've not tried that. So that's the reason I have not done it, is because I worry that the fluorescence may interfere somewhat with the gas exchange measurements when they're collected that rapidly. So I've not tried it. I've just been fearful of doing it. So feel free to see if it works.
All right. Does anyone else have any other questions? We do. Is there or will there be an application note describing these methods?
There hopefully is. It may not be, it describes them to some extent, probably. I don't know if it's adequate, but yes, there is one already on the website.
Maybe we can take a look at that, Jim, and expand on it if you feel we need to.
Yeah, I haven't looked at it in quite a while, but yeah.
Okay.
Sure.
Moving on. What is the standard reference of ACI value to compare with treated samples?
I guess I'm not really understanding that question. You know, ACI curves respond to environmental or treatments just like anything else. So if you have a plant that's been treated with with a high temperature stress or a water stress, it'll have a very different ACI curve than a control. But you know, that's fairly standard.
Mm-hmm.
And what you can do is you can you can make estimates of Mesophyll conductance for your treated versus your control and see if that's part of the difference in assimilation rate. But yeah, I wouldn't, you would expect that any stress is going to affect the shape of an ACI curve. And it may or may not affect Mesophyll conductance. Yeah, if I've misunderstood that question, just rephrase it and I'll try again.
All right. Yeah, I mean, I'm sure we can follow up afterwards, Jim, and
yeah,
try to get some clarification. We'll give one last call for any additional questions. If not, I think we'll end there. So again, I'd like to thank everybody for attending this webinar. It'll be made available on our website, www.ppsystems.com, probably within the next week or two. So feel free to check back in. If you think of any other questions or any other unrelated questions that you may have for PP Systems or Dr. Bunce, please feel free to email us and we'll be glad to respond to any questions. Otherwise, thank you very much, everyone.
So, oh, that's just a thank you. So one thing I just
done the,
what's that, Tim?
Unless you've got something to add.
I do. So what I did want to say is I put in the chat the link to our Mesophyll Conductance application note. And Jim and I will take a look at it and see if he feels there's something that he'd like to add. I did want to thank Jim again for his presentation and to everyone for your questions. And as Tim said, if you have anything related to this material, feel free to reply to the email I had sent you with your contact information for the webinar. If you have any unrelated questions, you can email support@ppsystems.com or ask the experts at ppsystems.com. And both email addresses will be in the follow-up email you'll receive from me with the link to this recording. That's about it.
Okay.
All right. Well, thank you, everyone.
Okay.
Thank you. And thanks again, Jim. Thanks, everyone. Yep.
Thank you for attending this webinar. To learn more about how the CIRAS-3 portable photosynthesis system can enhance your research experience, visit ppsystems.com.